You can write the text, the material, and after the class, I sent the PDF file of my lecture for you in WhatsApp in WhatsApp group. History of microbiology. First scientists in microbiology in discovery of microorganisms was Anthony van Leeuwenhoek. Anthony van Leeuwenhoek. Anthony van Leeuwenhoek was the first person to observe living microbes. Anthony van Leeuwenhoek was the first person to observe living microbes. He observed living organisms via single lens, single microscope, elementary microscope, initial microscope, single lens. Single lens magnified up to 300x. Single lens that he used magnified up to 300x. Anthony van Leeuwenhoek probably was the first person to observe living cells with a simple microscope. Amateur scientists ground his own lenses and described what we know today as bacteria, rod-shaped, spiral-shaped, etc., animalcules. It means he observed some organisms via single microscope. He observed some organisms. Some of them are motile, and he named them animalcules, not microbes. At that time, he named the organisms animalcules. Some of animalcules that he observed was rod-shaped. Some of them were spiral-shaped. Some of them were motile, and etc. They named them animalcule. This image shows Leeuwenhoek's work. Single lens, a specimen holder, focus tube, handles. The conflict over spontaneous generation. Some of scientists, they believe to spontaneous generation. They think living organisms origins from non-living things. Spontaneous generation. Spontaneous generation means living organisms can develop from non-living or decomposing matter. Spontaneous generation says living organisms can develop from non-living or decomposing matter living organisms can develop from non-living or decomposing matter. But some scientists, they studied more and more and found the spontaneous generation is not true. It's wrong. Living organisms can originate from living organisms. Life originates from life. Life originates from lives. For example, Francisco Redi disproved spontaneous generation for large animals. Francisco Redi disproved spontaneous generation for large animals. And he showed that maggots on decaying meat came from fly eggs, and showed that maggots on decaying meat came from fly eggs. Francisco Redi opposed the prevailing theory of spontaneous generation. Francisco Redi opposed the prevailing theory of spontaneous generation. He examined the examinations. He had the experiments. He did the experiments. Maggots in meat. He used covered jars to show that maggots came from files. Strong evidence against spontaneous generation. He used covered jars, yes, for preventing of microorganisms. He used covered jars to show that maggots came from flies, like mites. Strong evidence against spontaneous generation. Nowadays, we speak about the theory of biogenesis. We speak about theory of biogenesis, not spontaneous generation. Spontaneous generation is not true. It's wrong. In theory of biogenesis, life comes from life. Life originates from life. Living organisms originates from another living organisms, not from non-living organisms. not from decomposing material. Okay? But issue of spontaneous generation was actively believed for many more years. But issue of spontaneous generation was actively believed for many more years. This picture shows Francisco ready tests meat. First, meat in open jars. The microorganisms, the flies can grow in it and this causes spoilage of meat. 2. The jars with sealed trap, tightly sealed jars with cap. And 3. Jars covered with cloth netting. But could spontaneous generation be true for microorganisms? But could spontaneous generation be true for microorganisms? Some of scientists studied and explained the results. For example, John Needham. What is his experiment? His experiment was mouton brass in flasks and then boiled and then sealed. Mouton brass is flasks and then boiled and sealed. Results. Finding. Their finding showed brass became cloudy and contained microorganisms. Results showed brass became cloudy and contained microorganisms. لازارو اسپلنزانی he did brass in flasks and sealed and then boiled. Brass in flasks and sealed and boiled. Results. No growth of microorganisms. No growth of microorganisms. No growth of microorganisms. Because at first it sealed the jar and then boiled. And then microorganisms couldn't grow. And they had no growth of microorganisms. What about the ability of air to generate life? What about ability of air to generate life? It was not allowed to enter. Some scientists studied more experiments about the presence of microorganisms in different places and the role of microorganisms in different phenomena. For example, Louis Pasteur is a famous person. He is a famous scientist in microbiology. He did many studies, many experiments, many investigations. Louis Pasteur placed nutrient solution in flasks. Placed nutrient solution in flasks. Created flasks with long curved necks and then boiled the solutions. Left flasks exposed to air. He placed nutrients solution in flasks, created flasks with long curved necks and boiled it. Left flasks exposed to air. Results. What was the results? The finding showed no growth of microorganisms. No growth of microorganisms. No growth of microorganisms. Excuse me, I come 10 late. 10 minutes. Louis Pasteur, I said you, Louis Pasteur placed nutrient solution in flasks, created flasks with long curved necks, boiled the solutions, left flasks exposed to air. Results: no growth of microorganisms. Final blow to theory of spontaneous generation. Final blow to theory of spontaneous generation. A famous scientist named Tyndall, John Tyndall, studied about microorganisms. We have a technique named Tyndallization. Tyndallization, like pasteurization, sterilization, Tyndallization. Tyndallization is a procedure for control of microorganisms to prevent of growth of microorganisms. I speak about Tyndallization, pasteurization, sterilization in next sessions. Tyndall demonstrated that dust carries microorganisms. Tyndall demonstrated that dust carries microorganisms and showed that if dust was absent, nutrient baths remained sterile, even if directly exposed to air. Tyndall demonstrated that dust carries microorganisms and showed that if dust was absent, nutrient baths remained sterile, no microorganisms, without microorganisms, even if directly exposed to air. Also provided evidence for the experience of exceptionally heat-resistant form of bacteria. Also provided evidence for the existence of exceptionally heat-resistant form of bacteria. More evidence, more experiments. Berkeley demonstrated that the great potato blight of Ireland was caused by a fungus. He demonstrated that the great potato blight of Ireland was caused by a fungus. And Heinrich Barry showed that smut and rust fungi caused cereal crop diseases. Heinrich Barry showed that smut and rust fungi caused cereal crop diseases. And Louis Pasteur described that the peprine disease of silkworms was caused by a protozoan. Louis Pasteur worked about bacteria, about yeast, about protozoan, about fermentation, about pasteurization, and he had more and more experiments about microorganisms. Alexander Fleming. You know Alexander Fleming. Most of people know about Alexander Fleming. Alexander Fleming was a Scottish physician and bacteriologist. Alexander Fleming, a Scottish physician and bacteriologist, observed mold growing on a bacterial culture. There was a ring of clearing around the mold where the bacteria didn't grow. The mold was later found to be a penicillium species, and the naturally secreted chemical was called penicillin, an antibiotic. Alexander Fleming worked with Staphylococcus, a genus of bacteria, Staphylococcus aureus, a gram-positive bacteria. He worked with Staphylococcus bacteria. One day he cultured Staphylococcus on culture media. and culture media polluted with a fungus. And he didn't pay attention and continued the work. Next day, he saw a round of the fungus colonies, the Staphylococcus aureus didn't grow. It was interesting for him. Thus, Alexander Fleming said and described a material, an antibacterial agent is secreting from fungus can prevent the growth of bacteria. He studied and studied and studied, and then he discovered the first antibiotic named penicillin from Penicillium notatum fungus. Yes. Alexander Fleming, a Scottish physician and bacteriologist, observed mold growing on a bacteria culture. There was a ring of clearing around the mold where the bacteria didn't grow. The mold was later found to be a Penicillium species, and the naturally secreted chemical was called penicillin and antibiotic. This picture is the growth of Staphylococcus on culture media and penicillium colonies and green colonies. Okay. Final proof. Robert Koch established the relationship between Bacillus anthracis and anthrax. Bacillus anthracis is a gram-positive bacteria, can cause anthrax in human and in animal. Anthrax is a zoonosis disease. It means it causes in both animal and humans. Robert Koch established the relationship between Bacillus anthracis and anthrax. Use criteria developed by his teacher Jacob Hennel. Use criteria developed by his teacher Jacob Hennel, his teacher. These criteria, known as Koch's postulates, and Koch postulates now are very, very important. still used today to establish the link between a particular microorganism and particular disease. Koch postulates related to a microorganism and infectious disease. Koch postulate says a microorganism can infect a human and can cause infectious disease, and we can isolate that microorganisms from that person and can culture it on culture media and isolate again. Yes. Koch postulates 4 items for Koch postulates. One, the microorganisms must be present in every case of the disease, but absent from healthy individuals. The microorganisms, the organisms must be infect a person, yes, and cause infectious disease. It is the first item. Please wait. Must be isolated and grown in a pure culture. The suspected microorganism must be isolated and grown in a pure culture. And grown in a pure culture. And 3, the same disease must result when the isolated microorganism is inoculated into a healthy host. The same disease must result when the isolated microorganism is inoculated into a healthy host. And 4, the same microorganisms must be isolated again from the diseased host, from the infected host. Thus, in first stage, a microorganism must be infect a person, and we can isolate the organisms from this person, from that person. And that organisms can infect another person. And we must to culture the organisms. And the same organisms must be isolated again from the infected hosts, infected person, diseased host. Koch's and Pasteur work, yes, both of them, Koch's and Pasteur works helped establish the germ theory of disease. Yes, they established germ theory of disease. It means the role of microorganisms. The role of microorganisms in different phenomena, in different reactions, but via Koch's and Pasteur's experiments, Koch's and Pasteur's investigations. Germ theory of disease says microorganisms cause disease in people, animals, and even plants. A microorganism, a bacteria, a fungi, a virus, a protozoan can cause disease in people or in animals or in plants. The development of techniques for studying microbial pathogens. The development of techniques for studying microbial pathogens. The development of techniques for studying microbial pathogens. Koch's work led to discovery or development of agar, petri dish, nutrient brass, and nutrient agar methods for isolating microorganisms. Yes. Koch works led to discovering of development agar. Agar used in culture media. I will teach about different culture media, basal media, differential media, selective media, biochemical media, and most of media that we use about culturing of microbes. Petri dish or plate, and culture media like nutrient brass, nutrient agar. Nutrient brass is brass. Nutrient agar is solid for observing of colonies. And brass media for growing of bacteria. And media is cloudy. Methods for isolating microorganisms. Techniques for organisms isolation. Immunological studies. Edward Jenner. Edward Jenner used a vaccination procedure to protect individuals from smallpox. Edward Jenner used a vaccination procedure to protect individuals from smallpox. Note: this preceded the work establishing the role of microorganisms in disease. Industrial microbiology and microbial ecology. Industrial microbiology and microbial ecology. I said you, Louis Pasteur, studied about the different branch of microbiology: medical microbiology, industrial microbiology, microbial ecology, and etc. So, Louis Pasteur demonstrated The alcohol fermentations and other fermentations were the result of microbial activity. Louis Pasteur demonstrated that alcohol fermentations and other fermentations were the result of microbial activity. Developed the process of pasteurization to preserve wine during storage. Yes. The first scientists about controlling of microorganisms was Louis Pasteur. He described pasteurization to prevent, to kill some of microorganisms. And this technique named pasteurization. Nowadays, we use pasteurization for milk, for vinegar, in another country for wine. Yes? Pasteurization for controlling growth of pathogenic microorganisms. I said I will speak about pasteurization, tyndallization, sterilization, and different methods about them in next sessions. Yes. Tree of life. Tree of life. The recent taxonomy, the recent classification, the recent taxonomy, the recent classification about all of organisms, about all of microorganisms is tree of life. Tree of life include three domains. Tree of life is three domains of organisms. First domain, bacteria. Bacteria. Bacteria include pathogenic bacteria, non-pathogenic bacteria, and photosynthetic bacteria, chemosynthetic bacteria. All of bacteria is related to domain bacteria. The second domain is archaea. I will speak about archaea. Archaea, unsheathed bacteria, archaea or archaebacteria, like Uri archaeota, Tom archaeota, Core archaeota, Nano archaeota, and etc. And three domain of life is eukaryotes. Three domains of life is eukaryotes. Eukaryotes include plants, green plants, animals, fungi, protozoa, helminths, and algae. Bacteria, archaea, and eukaryotes have common ancestor. Yes, common ancestor. Tree of life have common ancestor. This picture is very interesting. Three domains. The highest, largest category, recent addition. Three domains. Archaea, bacteria, eukarya. Archaea means ancient bacteria. Archaea means ancient bacteria or archaebacteria. Archaea are prokaryotes. Archaea are unicellular, like bacteria. Unicellular like bacteria. Also simple cell structure. Also simple cell structure. Prokaryote, no nucleus, but have distinct metabolism. Chemistry. Allowing them to exist in extreme environments. Allowing them to exist in extreme environments. 2. Bacteria. Bacteria are unicellular, prokaryotes, and found in everywhere, in water, in soil, in air, in your intestine, in your mouth, in your skin, in your plant, on plant, in everywhere. We can search bacteria, we can see bacteria. Old kingdom named Munera. 3. Eukarya. Eukarya are unicellular or multicellular. Most of them are multicellular. And some of them are unicellular. Complex and organized cells with nuclei and organelles. Mitochondria. Unicellular to multicellular, complex and organized cells with nuclei and organelles. Mitochondria. Bacteria and archaea don't have mitochondria, but eukarya have mitochondria. Tree of life, yes, in another picture. Bacteria, gram positive, gram negative, photosynthetic, chemosynthetic, pathogenic, non-pathogenic. Archaea, like core archaeota, shrine archaeota, Uri archaeota, Tom archaeota, Nano archaeota, and other. And eukarya, animals, fungi, ciliata, flagellata, slime molds, and Cell means algae and other. About domain eukarya. The cells of all eukaryotes have a membrane-bound nucleus. The cells of all eukaryotes have a membrane-bound nucleus. Membrane of the domain eukarya are further categorized into one of four kingdoms. Four eukarya kingdoms include protista, fungi, plants, and animals. Yes. Four eukarya kingdoms are protista, fungi, plants, and animals. For any of microorganisms, we have a category of classification. Category of classification include domain, after that kingdom, phylum, class, order, family, genus, species. And after a species, we have a strain. Each species can have some strains. Genus, each genus can have many species, and one species can have many strains. Yes, domain, kingdom, phylum, class, order, family, genus, and species. In another slide. Most inclusive, least inclusive. Ten minutes rest. A bacteria, rod bacteria, rod or bacilli bacteria. You can see cell wall, the external layer of bacteria, cell wall. Some of bacteria have capsule or a slime layer on cell wall, and some of bacteria don't have capsule. cell wall, cell membrane. Cell membrane surrounded cytoplasm. The composition of cell wall in different bacteria is variable, is different. But the composition of cell membrane is same. Cytoplasm. Cytoplasm include chromosome, RNA and DNA. Ribosome. Ribosome for protein synthesis and inclusion bodies, inclusion granules. Some inclusions in bacteria are for energy, are the source of energy. For example, for Corynebacterium, inclusions are the source of energy. After cell wall, some of bacteria have motility. They are motile. Some of bacteria are motile. They can move with flagellum. With flagellum. With flagellum. Some of bacteria have pilus or pili or fimbriae. This section speaks about fimbriae and pilus. Bacterial morphology arrangement. 1, 2, 3, 4. In this slide, 1 and 2. 1 form. 1 form is rod or bacilli. Yes, rod and bacilli or bacilli. For example, streptobacilli or bacilli. Like Bacillus species, Clostridium species, Escherichia E. coli are Bacillus form. 2. Cocci. 2. Cocci. Cocci is plural. Single form is cucus. Cucus, cocci. Different forms of cocci in bacteria are A. Cocci, single cocci. Single cocci. Individual cocci. B. Diplococci. Two cocci. Diplococci. For example, Neisseria meningitidis. Neisseria meningitidis. C. Streptococci. Streptococci. For example, Streptococcus pyogenes. Streptococcus pyogenes. D. Staphylococci. Staphylococci. For example, Staphylococcus aureus. Staphylococcus aureus. or Staphylococcus epidermidis or Staphylococcus saprophyticus. E. Sarcina, 4 coccus, Sarcina. And F. Tetrads, 8 coccus, tetrads. Micrococcus species, like Micrococcus species. Excuse me, Sarcina are tube in form, 8 cocci, and tetrad are 4 cocci. In this picture, this is single cocci, a coccus. Division in one plan. Diplococci, 2 cells. Another form, streptococci, cocci in chain, single cocci coccus. Tetrad, 4 cocci, and Sarcina, 8 or more cocci. Sometimes division in different plants, and it causes different morphology. Streptococcus, Staphylococcus, coccus in cluster means Staphylococcus. Coccus in pair means Streptococcus. Streptococcus in pair, in Streptococcus in chain, long chain or short chain, Streptococcus. One coccus, a coccus, is single, cocci is plural. Rod or bacillus. Bacillus is single, bacilli is plural form. Spirillum, spirochetes, and different forms. Other forms are filamentous, like fungi, like streptomyces, actinomyces are filamentous form. Another forms, hyphae form or stalk form, budding and appendaged bacteria. This picture, Diplococcus. Yes, Diplococcus. Single coccus and Diplococcus. Diplococcus. Rod or bacilli form. Rod or bacilli form. 3. I spoke about cocci form and rod form. Now, spiral. Yes, spiral. A spiral A, B, C. Vibrio, spirillum, spirochetes. Vibrio, spirillum, and spirochetes. You saw the related picture in last slides. In previous slides. Borrelia, spirochete. Borrelia, Borrelia, spirochete. Bacterial cell structure. Bacterial cell structure. Yes. Some of bacteria have appendages. Some of bacteria have appendages. And some of them, they don't have. What are appendages? Appendages like flagella, pili, or fimbriae. Appendages like flagella, pili, or fimbriae. Surface layers like capsule, cell wall, and cell membrane. Cytoplasm. Cytoplasm include nuclear material, ribosome, mesosome, and inclusions. Nuclear material like RNA and DNA. Ribosome for protein synthesis. Mesosome for DNA replication and respiratory reactions. And inclusions for source of energy and other things. And special structure like endospore. Some genus of bacteria have endospore. Some genus of bacteria have endospore. They are endospore forming bacteria, such as Bacillus sp, Bacillus species, Clostridium species. Both Bacillus and Clostridium species can produce a spore form in unsuitable environment, in unsuitable conditions. We speak about endospore. We will speak about endospore. Appendages. One flagella. One flagella. I told you some of bacteria have flagella because some of bacteria are motile. All of bacteria are not motile. Yes. Some of rods or bacilli bacteria and spiral bacteria have motility. They are motile. Motile. Their function is motility, means move to another places. Origin. Flagella originates from cell membrane. Yes. Flagella, pili, fimbriae, all of them originates from CM, cytoplasmic membrane or cell membrane. Origin of flagella is cell membrane. Cell membrane flagella attach to the cell by hook and basal body, which consists of sets of rings and rods. Yes. Cell membrane flagella attach, connect, attached or bound to cell by hook and basal body, which consists of set of rings and rods. In next slide, I will show you the structure of flagella. Structure of flagella in gram-positive bacteria and gram-negative bacteria is different. In gram-negative bacteria, attach or connect or bound. Attach. Attach means invasion. Attach means connect. In gram-negative bacteria, two sets of ring and rods, L, P, S, M rings and rods. For example, E. coli, Escherichia coli. In gram-negative bacterium like E. coli, like Salmonella, like Shigella, like Neisseria, like Proteus, in gram-negative bacteria, two sets of ring and rods. We have L, P, S, M ring. But in gram-positive bacteria, like Bacillus megatherium, like Staphylococcus, we have S and M ring. S and M ring and rods. Structure of flagellum. Structure of flagellum. You can see the structure of flagellum. This is cytoplasmic membrane. Cytoplasmic membrane. After that, periplasm. Peptidoglycan is in periplasm. And after that, outer membrane. This is a structure of gram-negative bacteria. In gram-negative bacteria. You can see the rings, L rings, P rings, SM rings, and motor protein, and motor, mot protein means flagellum made in protein named flagellin. Flagellin. Flagella. The function of flagella is motility, yes, or movement. Some of bacteria can swarm on culture media. This movement named swarming. Swarming occurs with some bacteria. Swarming means bacteria can spread all of culture media on different part of culture media. Spread across petri dish, such as Proteus vulgaris, and Proteus mirabilis can swarm on culture media, can spread across petri dish. Arrangement basis for classification. Arrangement basis for classification. Monotrichus, one flagella. When a bacteria have one flagella, it means monotrichus. Lophotrichus, tuft at one end. Cophotrichus, tuft at both ends. Amphitrichus, both ends. And Peritrichus, all around bacteria. When the flagellum spread all of the surface of bacteria, Peritrichus. Peritrichus bacteria like E. coli, Escherichia coli, like Proteus, like Salmonella. Some picture. Monotrichus, Amphitrichus, Lophotrichus, and Peritrichus. The structure of bacterial flagella allows it to spin like a propeller and thereby propel the bacterial cell clockwise or counterclockwise wave-like motion. Yes, two type of movement, clockwise or counterclockwise wave-like motion. Bacterial flagella provides the bacterium with mechanism for swimming toward or away from chemical stimuli. A behavior is known as chemotaxis. Chemotaxis. Sometimes a bacteria go to attractive agents and sometimes no repellent. This phenomenon named chemotaxis. Chemotaxis. Chemotaxis related to the type of movement of bacteria. Chemotaxis is a behavior related to movement of bacteria. A behavior is known as chemotaxis. Chemosensors in the cell envelope can detect certain chemicals and signal the flagella to respond. The structure. I spoke about structure. I told you flagella made by protein named flagellin. Yes, protein in nature, subunit flagellin, globular protein. Flagella movement. In this picture and another picture, you can see the type of movement, the type of chemotaxis. For Pitricus bacteria, two type of movement, two type of rotation, CCV rotation or CCW rotation or CW rotation. In CCW rotation, bundle flagella and move to attractant. In CW rotation, the flagella not bundled. Tumble flagella pushed apart. And in this page, in this picture, for polar flagella, you can see the type of rotation in CCW rotation or in CW rotation. Fimbriae and pili. Another appendages in bacteria is fimbriae or pili. What is the difference between fimbriae and pili? Fimbriae is common pili. Fimbriae is common pili. The role of it is attachment or adherence, connect to cell wall, connect to epithelium cell of intestinal tract, epithelium cell of respiratory tract, of urogenital tract, and fimbriae is important factor for pathogenicity, for virulence of bacteria. What pili means 6 pili? It used for conjugation. Conjugation is a way for transferring genetic material from a bacteria to another bacteria. I teach about transmission of genetic material in next sessions. Fimbria and pili. Fimbria. Fimbria is shorter than flagella and straighter, smaller, hair-like appendages. Yes. Fimbria is shorter than flagella and straighter, smaller, hair-like appendages, only on some gram-negative bacteria, like E. coli, like Proteus. Function. The function of Fimbria is adhere, is attachment, not involved in motility. Motility is related to flagella. Fimbria or pili is not related to motility. It's related to adherence. Yes. One of the invasive mechanisms of bacteria. Some pathogenic bacteria have pili or Fimbria, and because of having pili, they can connect or they can adhere to mucosal epithelium, to epithelium of intestinal tract, epithelium of orogenital tract, epithelium of respiratory tract, and other things. Some pathogens cause diseases due to this antigenic characteristic. Prevent phagocytosis. Yes, in some bacteria, pili or Fimbria related to phagocytosis. Their capacity, their effect is anti-phagocytic activity. Yes, anti-phagocytic activity. They are anti-phagocytic agent. Means prevent, they can prevent from phagocytosis. Pili means 6 pili. It is necessary for bacterial conjugation. Yes, transfer of plasmid or genetic material from a bacteria to another bacteria. Pili 6 factor. If they make pili, they are positive or donors of F factor. F means fertility. F factor means fertility factor. It's about conjugation. It is necessary for bacterial conjugation, resulting in the transfer of DNA from one cell to another. I have been implicated in the ability of bacteria to recognize specific receptor sites on the host cell membrane. It has been implicated in the ability of bacteria to recognize specific receptor sites on the host cell membrane. B. Origin. Yes, origin. Origin of flagella, origin of fimbriae, origin of pili is cytoplasmic membrane, cell membrane. Yes, in all of them, origin is cell membrane. And the composition of them is protein. For flagella, flagellin. For pili, pilin protein. Position. Common pili numerous over the cell, usually called sex pile, 1 to 4 per cell. Structure. Composed of proteins which can be dissociated into smaller unit pilin. Yes, about flagella we told flagellin. About pilin is pilin. About pili is pilin. Pilin and flagellin are protein. It belongs to a class of protein, lectin, which bond to the cell surface, polysaccharide. Cell surface layer. Cell surface, one slide. I have one slide. Cell surface layer. Glycocalyx. Yes, some of bacteria have glycocalyx. Means capsule or slime layer. All of bacteria don't have glycocalyx, don't have capsule. Many bacteria are able to secrete material that adheres to the bacterial cell, but is actually external to the cell. The composition of glycocalyx or capsule is different in different bacteria. In most of bacteria is polysaccharide. In some bacteria is polypeptide. It consists of polypeptide and polysaccharide on bacilli. Most of them have only polysaccharide like streptococci, and it is a protective layer that resists host phagocytosis. Yes, capsule have antiphagocytic activity. Medically important, Streptococcus pneumoniae. Capsule is the most pathogenic factor, the most pathogenic agent for Streptococcus pneumoniae. Streptococcus pneumoniae cause pneumonia in human. Don't tire. Thank you a lot. Thanks a lot. Good evening. If you have a question, you can send me message via WhatsApp group or in this session.